The basic idea of Punnett square is to start with the idea of meiosis. If you remember a little bit about meiosis from 140, this will be a little review. If you don't, we'll cover meiosis in a little bit, but just as a preview. The idea of the principle of segregation is that the two factors, in this case capital R and lowercase r, will be separated from each other when you go through the process of making the cells that we call gametes. And just make sure that everything is more or less correct. The first step is always the duplication of DNA, and then we have a first division step where we separate one piece of DNA from another piece of DNA, and then we go through another round of cell division where we separate the remaining DNA molecules from each other. And a Punnett square is just a shorthand way to look at what are the possibilities of alleles that an individual could give to their offspring. Remember, Gregor Mendel started to experiment with true breeding parents, so it's what we call homozygous. In a homozygous, you have two copies of the exact same allele. In this case, we just use capital R and capital R for one true breeding parent and lowercase r for the other true breeding parent. At the beginning of the experiment, you don't really know what is dominant or recessive, but going through the experiment, you can determine what is dominant or recessive. But in this case, we're just sort of assuming things, or just saying that this is dominant, this is recessive, just for simplicity's sake. And here we see what is going on directly in the making the F1 generation. If you don't remember anything about Punnett squares, you have the parent genotype, or sorry, gametes, across one side and across the other, and then inside the box is the offsprings. Basically add the two things together, capital R plus little r gives that one, capital R plus little r gives that one, and so forth. And if you look at this, when we take true breeding parents and mix them together, we get offspring that are all exactly the same. And that's the important thing about Gregor Mendel's work in relationship to the idea that we call complete dominance, is everything in the F1 looks exactly the same. But more importantly, to remember about Gregor Mendel's work, and as we go forward looking at the idea of complete dominance, all of the offspring look like one parent and not the other. And all of the F1 offspring will look like the dominant parent and not the recessive parent. But the recessive allele is still there. It's not gone or changed, it's just sitting there, not being able to be observed. And so if we take these individuals and crossbreed them to themselves or to another F1 individual, we get a Punnett square that looks like this down here, where now we can see three of our individuals are now going to have the dominant phenotype and one will have the recessive phenotype. But the Punnett square illustrates that genotype and phenotype don't always mix or match each other. So we have here a molecular phenotype. Three of them are purple, one of them is white, in the case of Gregor Mendel's experiment. A molecular trait, we have homozygous dominant, homozygous recessive, and two things that we call heterozygote. So we do see that sometimes it could be a match morphologically, but not molecularly. Molecularly. I'm gonna keep trying to practice molecularly. I should say molecularly. This important thing to keep in mind is genotype and phenotype don't always tell the same story. In this case, with complete dominance, a capital R is enough to give you whatever the dominant trait is, purple or yellow or green or wrinkled or whatever it is. But the principle of segregation says that when parents breed together, they separate their factors into cells and give those factors to their offsprings individually. So you either get one or you get the other. And the principle of segregation was simply based off of doing the monohybrid crosses, which is, this is basically an example of monohybrid cross. They're only different in one trait. But to look at a dihybrid cross, we're looking at parents that differ in two different traits. And the example that is commonly used is this one here, where you have a yellow round and a green wrinkled. As the true breeding parents, you breed these together and all of your offspring in the F1 are yellow round. Same basic pattern that we saw with a monohybrid cross. all the F1 offspring look exactly the same and match one parent, but not the other parent. So the idea of dominant versus recessive is still played out here. If you take these F1 offspring and you breed them to themselves or to another F1 individual, you get what is now called a hybrid cross, so our ratio is going to change a little bit. Instead of a 2x2, we get a 4x4 little Punnett square matrix. And we'll deal with how we sort of separate these things. But one of the more important things about looking at the dihybrid cross is Mendel's principle of independent assortment. What the independent assortment principle said was the factors that control one trait are not affected by the factors affecting another trait. So the inheritance of yellow or green is completely independent of the inheritance of round versus wrinkled. If they weren't, then you would never see something where you have green and round. Because if they were connected, the green and wrinkled would always be together regardless of what you did. Same thing would be true with yellow and round. They would always be together if they were inherited together as a unit, or if they were dependent on each other. But because you can mix and match these, or at least you see the mixing and matching of the two different phenotypes in our offspring, we do know that their two things are independent from each other. But the cool thing about the dihybrid cross is the 3-to-1 ratio is still in here. It's just now more of a binomial equation. 3 plus 1 squared gives us 9, 3, 3, and 1. But with any phenotype you look at, whether it is 2 together or 1 separately, there's always a 3-to-1 ratio. So if that recessive phenotype is showing up at 25%, regardless of how you look up the numbers. If you just look at colors, there's 12 to 1, 4 of another, again, a 3-to-1 ratio. What it looks like, smooth versus wrinkled, again, a 3-to-1 ratio. And combining two together is always 3-to-1. So again, this idea of 3-to-1 is an important idea of the F2 ratio in complete dominance. It's not important to remember the specific examples here. We're going to do problems with Punnett squares that are more important to try and memorize this example. But using this example, we have illustrations of the principle of segregation and the principle of independent assortment. Again, it says law, but I don't really care. Law, principle, it's probably a little better to say principle because that's really what Gregor Mendel used. But as I mentioned earlier, what it says independent assortment. The factors for different traits are independent of each other. Unless they're not. Because there are exceptions to these things. And Gregor Mendel was very lucky. Or very shrewd. Or over 8 years, he was able to plan out things really well. And as I mentioned on Wednesday, some of the data he got didn't really fall into the pattern that the story he was trying to tell. And so sort of ignored that data for publication. It's not something it's not an unknown thing. Scientists are known to take the majority of what happens rather than the exceptions. But things can be independent, and usually are, but we'll get into an exception to that. But more importantly, as we talk about meiosis, we'll talk about the mechanisms that actually make traits independent of each other and separatable in offspring. So all of that was basically trying to get us to the point where we can talk about complete dominance and other patterns of dominance and things that don't fit into a very nice and neat story. So if you haven't really been paying attention, we're going to give us some important sort of properties or characteristics or facts that relate to the different types of interactions we see in inheritance patterns. And start with the very first thing that's most important about Gregor Mendel's work is he was only looking at simple traits. Traits that are controlled by a single gene. So the inheritance of that gene is hopefully independent and not affected by other things. And he was also mainly focused on things that had a clear dominant, recessive, or difference between one phenotype and another phenotype in the other parent. And as we're going to learn, complete dominance is not the only one. I would like to argue that complete dominance is a rarity, and the more common thing is the weird things that we're going to talk about. But I might be wrong. So when we talk about complete dominance, there is a particular thing that is important to talk about. What makes recessive recessive? Is it just that purple is much darker than white and you never see any white color because of the darkness and the strength of the purple color? That's a possibility, but it's not the correct thing for complete dominance. What we typically see in complete dominance is that the recessive allele has a mutation that makes that gene completely inactive, or makes a protein that is inactive. So you have a functional protein making purple color, and a non-functional protein making no color. How we get this sort of purple versus white dichotomy. And so in the heterozygous example, purple protein makes purple, white protein makes nothing, so you get purple. There's no mixing of anything whatsoever. And that's not the case for other things, but it will be the case here with complete dominance. We typically see a true functional protein, 100%, zero functional sort of difference between dominant and recessive.
So the recessive allele isn't necessarily white, it's just a lack of purple?
That is one way to think about it. And that's not true for every case, but for a simplistic model, the gene that controls flower color makes a protein that makes purple, pigment, whatever. But if you inactivate that protein, now it's no longer able to make purple, it makes white. And maybe it's a biochemical pathway where there's several steps, where you have to take A, turn it into B, then turn it into C, then turn it into D, and that last thing is actually the thing that's purple. Maybe one of the genes is broken in the steps, so you can't complete the process. So can get more complicated than simply this protein doesn't work, therefore you don't get purple color. But for us, that thing is not just sort of this is generally what happens in the idea of complete dominance. How you cannot see something when it's still there. This is an easily explained possibility. Again, this is true for morphological traits. Again, molecularly, two of these are heterozygous, so that R, the little r, is still in there. It's not doing anything, but it will show up if you get two of those little r's together. And so typically when we talk about complete dominance, the F1 generation is not as important as the F2 generation. So phenotypically speaking, an important characteristic of complete dominance inheritance patterns is in an F2 generation, you always see a 3-to-1 ratio of a dominant trait or phenotype to recessive phenotype. Complete dominance, all of the F1 offspring are going to be exactly the same, and one of the things that makes complete dominance complete is the fact that all of the F1 offspring are the same as one parent or the other, not a mixing of those two colors together. That's the other thing we'll talk about in a second, where things actually do mix. But here, purple is active, white is inactive, or at least you have active and inactive. Active makes purple, inactive makes no purple, and so you get no color. So white is sort of the default color we get. There are only two phenotypes, in case we typically see with Gregor Mendel's work, purple or white, yellow or green. There's only two possibilities, recessive or dominant. So it limits the possibilities of what we get when you can only see complete dominance. Remember, F1 generation is all a heterozygous genotype. Now also, genotypically speaking, we can talk about complete dominance another way. And phenotypically, we see a 3-to-1 ratio, but for the genotypes of the offspring, we will want to think about, with complete dominance, we have a 1:2:1 genotypic ratio. And one of them was the homozygous dominant, the other one was the homozygous recessive, and the two were the heterozygous. If you don't believe me, you can go back to this one Punnett square slide. Forwards and backwards. But here again, when we look at Punnett squares, we know that two of the individuals have a different genotype, even though they have the same phenotype. And so here again, we see a molecular and a morphological difference in complete dominance. And I again want to stress that with complete dominance, the molecular and morphological is separated. We see a difference in what's going on. And why do I make such a big deal about this? Because in another one, it's not the case. And it sort of helps to think about things. As I've kind of been talking around, it turns out that in some of the people that have looked at Gregor Mendel's work, and I have read it because I can't speak German or read German for that matter. Anyways, some of the people that have analyzed Gregor Mendel's unpublished results think that maybe some of the things that he looked at that didn't fit his pattern probably fall into the category that we now call incomplete dominance. So we have complete dominance and incomplete dominance. That's so fun. So with incomplete dominance, we see a different type of pattern. And that's not something I need you to remember, but when it comes to incomplete dominance, there is a case where it is observed more often. And what a lot of people, or a lot of literature says that in flowering plants, incomplete dominance is very common. Gregor Mendel didn't see it. So a pea plant makes flowers, but it's not a flowering plant. Because it's a vegetable or, I don't know, what are you gonna call it? Peas or tobaccos. Everything makes flowers, right? That's how they reproduce. But a rose is a rose because it's just a rose, or a lilac, dandelion. Not making something edible, and it's totally dandelion leaves. Anyways, I digress. When we look at incomplete dominance, instead of there being a complete or a complete dominant and a recessive, there's an intermediate state. So right off the bat, with incomplete dominance, we are going to see three phenotypes for a particular trait, in this case, flower color. But unlike in every experiment, you always start with true breeding parents. So in this case, we'll start with a plant that makes red flowers and a plant that makes white flowers and breed them together. And if this were complete dominance, then we should get all red. But because this is not incomplete dominance, we get something that's in the middle, where we get pink. So the idea here is instead of having an inactive protein, you have two active proteins. One actively making red, one actively making white. And anytime you mix red and white together, you get pink. That's how you make pink paint. Take red and white and mix them together. And they mix it together really well. The problem here is, how do you determine what's dominant and recessive? Because both red and white disappear, only reappear, so is pink dominant and red and white are recessive? If you follow the pattern we had for complete dominance, that might be the conclusion. But just like in the earlier experiments, we need to go forward and do the F2 generation. That's where things really get fun. We take a pink flower plant and breed it to itself. Now you get red, pink, and white offspring. And so it's no longer a 3-to-1 phenotypic ratio. It's a 1-to-2-to-1 phenotypic ratio. But the cool thing about incomplete dominance, the genotypic ratio and the phenotypic ratio, the molecular and the morphological actually correspond with each other. Homozygous dominant is one thing, heterozygous is different, homozygous recessive is a third separate thing. That's kind of cool. And these are sort of, you know, red is always true breeding, white is always true breeding, which is kind of cool. But our F1 are always an intermediate state. This is why a lot of people sort of got the idea of blending of factors or traits originally. Because you can see it happen. Red and white make pink, purple and white make lavender. Pick your colors of white and make something that goes in the middle, I don't know. Nobody? Black and white make gray. What did he explain?
White, brown, and tan.
What?
White, brown, and tan.
I was gonna say orange and white make a creamsicle, but that's on topic. That's a fun joke though. You can use that, but you use that. But again, what defines and separates complete dominance and incomplete dominance is how the dominant and the recessive allele interact with each other. Again, with this, how do we determine what is dominant and recessive? Probably somebody arbitrarily said that red is dominant and white is recessive just because that's the pattern we saw for complete dominance. But I would argue, why can't white be dominant and red be recessive? Who says that making color has to be the dominant trait? Why can't it be the other way? But it's kind of an arbitrary determination which way you go with these things. We do see some clear differences. The one sort of similarity is the F1 generation in both are all the same color. It's just in complete dominance, one of that color is one of the parents, and with incomplete dominance, that color is in the middle, intermediate color between what the two parent colors are. And obviously, the phenotypic ratio for your F1 offspring is different. But the genotypic ratio is exactly the same. So it's one place we are going to see the same type of genotypic ratio. Except with complete dominance, the genotype and the phenotype ratios don't match, but for incomplete dominance, again, they do match. And unfortunately, I kind of asked you questions before we sort of even come up to the slide, but I was going to get you to try to think about how do you sort of answer these things. Does anybody have any thoughts?
I feel like you can't say one is dominant and one is recessive. Like, I feel like there's no conclusive way to determine it because you could argue either direction.
Yeah.
Could it be they're both dominant?
That is a possibility as well. They are, if they're both there in the same sort of percentage or ratio, or like it's 1-1 ratio, then they're both dominant and they're both recessive. Or because they're neither. But you're not seeing one being stronger or better than the other. We call it incomplete dominance, but really there might not be any true dominance going on. But they're just both there. Now if we go back to our little pea plant thing again, there are some other kind of physiological things that can be looked at. And again, the idea is codominance, we'll talk about in a second, sort of both being there, sort of independently and equally. Incomplete dominance is an intermediate state. And so if you look at this being the recessive and this being the dominant, and the thing in the middle, does that look like it's a mixture of these two things, or half of one and none of the other? Sometimes things get really confusing. What are we actually seeing? Yeah, I don't want you to worry too much about trying to answer this. But after we look at codominance, we can come back to this, look at this. Do I think this is incomplete dominance? Or do I think this is a codominance example? Again, if we go back to this one little picture that I had, oh, it's not on this set of slides, it's not. Yeah. Oh well, we won't go look at that graph. Sometimes it's not quite so easy to see things. But one thing that is much, or does make things much easier to distinguish is the other idea we talked about. Morphological traits tend to exhibit complete or incomplete dominance. Molecular traits tend to have a codominant example. And so would you argue that this is a molecular or morphological trait? Are we looking at DNA sequence right here? It has to be morphological. Just by definition, if we're not looking at the actual DNA sequence, then we're not looking at a molecular phenotype. Now I say that, but there are some, doesn't always fit. Just doesn't mean DNA exactly. But typically with molecular, we are looking at something that is not a observable phenotype exactly, but a DNA or a protein difference that is not resulting in a functional change. It's kind of hard to explain sometimes. Now, one of the first things I think we need to say about the last form of dominance inheritance pattern is that this is actually complete dominance with an extra dominant. So we should call it double dominant. Because it's going to obey a dominant recessive. It has nothing to do with incomplete dominance, but has a lot to do with complete dominance. But again, what makes this really different than complete dominance is the presence of multiple dominant alleles and at least one recessive allele. So dominant is always going to be completely dominant over recessive, but dominant and dominant, there is no winning because they're both going to be there. So how do you get both things present? Color-wise, there are some weird ways to do it. But blood type is typically used as the main example of codominance because we can physically see the presence of two different things being there. It's not a mixture of the two together. It's two separate molecules being present. And so if you don't know anything about blood type, we have A, B, B, O, A, B, O, A, B. 4 different blood types. And blood type differences are because of different sugars that are put onto the outside of the red blood cells. And so the absence of a sugar is O. In this case, O is a recessive trait. It's a non-functional protein. A makes a sugar and B makes a sugar. So if you're A blood, you make the sugar that is associated with A. If you're B, you make the sugar that's associated with B. That's why you can't give B blood to A people because they say that's not me. Because antibodies attack the B blood because they're used to seeing A blood. But if you're AB blood, you have A sugar and B sugar on your red blood cells. So you have both. They're equally and not mixing together. The idea of non-mixing is what really separates codominance from incomplete dominance. We observe both without a mixing or an intermediate state whatsoever. For whatever reason, the gene for blood type is called I. And they use superscripts of A and B to denote the different types of dominant alleles that are out there. Now, Gary, remember this? I'm never going to ask you how many phenotypes and how many genotypes there are for blood type. But I do use blood type with one little calculation we do later on in the semester, so it's sometimes helpful just to know what you're looking at. And so again, if you see capital IA here, that's the homozygous situation. Here is also a homozygous situation. Here is also homozygous. But these 3 here are heterozygous examples. Here we see dominant recessive and dominant and recessive, but this one is 2 dominants together. Typically, the lowercase I is used to again promote the idea of dominant and recessive between A and B and O. Some people just use IO so they can remember that little I is the O. You don't have to worry about that again. I'm not gonna also ask you this molecular information down at the bottom, I thought was fun and interesting, which is why I'm showing it to you. But the gene or the protein made from the A allele and the B allele is almost 99% identical. There are only 4 amino acid differences. But those 4 amino acid differences are enough to make the protein add 1 sugar or a different sugar. That's enough to give you one blood type versus another blood type. And incidentally, what makes this recessive is a deletion, which creates a frame shift. So it's a non-functional protein produced from this gene. So you don't make any sugar at all, or don't attach any sugars. That's why it's sort of missing something. And blood type is not the only example that we see. I have a few examples to show you in just a second. And again, what truly separates codominance from incomplete dominance is there's no blending of things, no intermediate state. You see both dominant alleles being expressed independently and distinct from each other. But A is completely dominant over O, B is also completely dominant over O, so if you would mix those together, you would get a typical 3-to-1 ratio. For the F2 generation, you can't have a predictable pattern, because you never know what your parents are going to look like from the very beginning. Does anybody know their blood type? So I've always find it weird people don't know their blood type. What are you going to do? I mean, if you're knocked out, who cares? You can't answer anyways. But typically, if you give blood, they typically will tell you what your blood type is. You don't know. Which is how I found out. You may give them blood and not been told what your blood type was. I mean, they did tell you what your blood type was because you gave blood. Okay. Cool. Now, blood cells are not the only time you see these weird types of examples. There are some fun ones. In some flowers, you can see codominance. So instead of a pink flower, you see individual petals of red versus white. So you see sort of a mosaic pattern, where sometimes you get red and sometimes you get white. Cows, there's also an example of complete dominance in cows. Who likes cows? Lynn thinks cows are cute.
They taste good.
They do taste good, but they're also kind of fun.
I think they're pretty.
You should watch the movie City Slickers. You'll fall in love with cows. City Slickers. Anyways, this is neither, it's not beige or tan, it's some brown, some white. There are weird situations where this isn't codominance. In some cats, like calico, the reason why you get yellow and black and white, it's not a codominant example, it's something else. So it's all sorts of weird things. I don't really know why people say that sickle cell anemia falls into codominance. But you either are a good red blood cell or you're not a good red blood cell. But that's what they say. I don't really believe it. We'll talk more about sickle cell anemia a little bit for some fun little stories. So basically, we just need to know some basic facts about what makes complete dominance between the phenotypes that we see, how often does a phenotype show up, compare that to incomplete dominance, and also sort of think about codominance and how it's linked to complete dominance. For complete dominance, we typically can see multiple recessive alleles. For incomplete dominance, it's usually not multiple recessive, it's just one dominant, one recessive. I tried to find an example of multiple dominant and multiple recessive, but I couldn't really find an example that's not really fit. Luckily for you, you'll never be asked to look at something and say, is this incomplete dominance or codominance? But we will have to separate complete dominance from incomplete dominance. So again, we're looking at incomplete dominance. The heterozygous genotype has a phenotype that's different than either parent. So you do see that third phenotype show up with incomplete dominance. Complete dominance only has two phenotypes to worry about. Anybody have any basic questions or general questions or burning questions or questions in general? Or questions in burning? Right now, before we move forward. I always ask the question, and hardly ever are there questions. So the question is, should I continue to ask? Are there any questions? Yes, I should, or no, I should?
I have a question, so yes.
Good.
So are there instances where the dominant is a non-functional protein and then the recessive is a functional protein?
I would say, without knowing, most examples of complete dominance are what they are, where the allele is dominant. It's dominant and one is recessive is not. In the case, if we look at diseases, there are what we call autosomal dominant and X-linked dominant disorders. And so there, it's the protein is malfunctioning, but it's still a dominant thing. And in those cases, typically the recessive is non-functional or weaker, so it's not causing the same disease state as the dominant allele is. But I don't think dominant would ever be non-functional, because functional should always trump non-functional. There might be exceptions, I just don't know, I don't know. Human genetics might not. Any other questions? Good question. Basic idea is that complete dominance, incomplete dominance, and codominance are examples of simple traits. One gene affecting a single phenotype or trait. We do have several different types of complex atoms. The two that are easiest to explain are pleiotropy and epistasis. The third one, variable gene expression, is a little bit harder to explain. It does come in two different varieties, what we call variable expressivity and penetrance. Again, remember with simple traits, one gene affecting one trait. 1-to-1. These are all examples of complex, so we have multiple things doing multiple things sometimes. And pleiotropy and epistasis are going to look a lot opposite from each other. So one way to think about it, it might make it easier to remember that pleiotropy is opposite of epistasis. But in all four cases, we do see that genotype and phenotype don't always mean what they mean or end up being the same. And so in all situations, your genotype might not be exactly, or give you the phenotype that you expect. For pleiotropy, the basic idea I want you to remember is that one gene affects multiple traits. Here we have some examples: schizophrenia, autism, sickle cell anemia. And so what you can kind of think about these things is one mutated protein might have multiple effects. And so if you think about autism, there are developmental disorders, there's also sometimes physical developmental disorders. Those two things are sort of separated from each other. Some are in communication skills, may vary, and that's just, we see some different things being affected. And it's again, the idea is one gene is having multiple effects on different traits. Not a simple trait, it's not 1-to-1, it's 1-to-3 or 1-to-5 or whatever. But some genes do have multiple functions or purposes inside organisms. Just as an example, we have this phenylketonuria, which is kind of fun to say. So it's a single gene And this gene is involved in taking phenylalanine and turning into tyrosine. Not you need to worry about memorizing that. But this inability to turn phenylalanine into tyrosine does have a lot of different effects on humans, from seizures, mental disorder, learning disabilities, behavioral problems, even making your urine smell worse than it already might. But think about how unrelated a seizure is to urine smelling bad. And the idea of one gene having multiple effects, and you can't predict all those effects. So it makes human genetics really hard to sort of work with. One mutation might have multiple effects you can't even account for. Now that I sit here and think about it, I should do epistasis next. But it's not the next slide. That would make more sense. I said they're opposites, let's take them together. But you don't. One of the most important things about the two different types of variable gene expression is the idea that it says a gene is variably expressed or expressed variably. So from one individual to the other, with the same genotype, the phenotype might not be the same thing because of the amount of gene expression we see in one individual versus the other individual. So same genotype, but phenotype can be different. Yes.
What causes the difference?
Things that we won't talk about. Any number of things could control the expression of a protein. As we'll go forward, we'll talk some about gene expression strategies that bacteria or prokaryotic organisms use versus eukaryotic organisms. A lot of times there's a protein controlling expression of another gene. And so you might have the same genotype for a gene, but maybe different over here. And that different over here is the reason why this difference here is now manifested. And this difference doesn't have an effect. This isn't over here, we will. And so there are like this idea of multiple things interact with each other, sort of everything, even though we try to put things in different boxes. I like polydactylism as an example of variable expressivity. Does anybody know Count Rugen? The six-fingered man from The Princess Bride? I've seen the movie The Princess Bride. Go watch it, it's a good movie. In it, there's a six-fingered man. Is six fingers rare or common? Rare. It's rare, right? Anybody have six fingers? We can just do the test for you right now. How common is six fingers in a group of 20 people? The thing that's weird about polydactylism, why it's sort of its variable gene expression, is sometimes you can have six fingers or six toes. You have six fingers on one hand, six toes on one foot, or you could have six fingers on both hands, or six toes on both feet. Or maybe you just have the trifecta, which is a quadfecta. You can have six digits on all of the appendages. That's the variableness to it. Anybody know what Marfan syndrome is?
Connective tissue. Is that where you just have like long limbs?
Yeah, typically it's associated or height. Being really tall is typically this, but it's sort of an in proportion tallness. I really only know it because in one of the movies with Vince Vaughn, someone calls him Marfan. What the hell are they talking about? Oh wait, it's a disease. Yeah, she's making fun of it.
So is that like gigantism, but not as extreme?
Well, again, the idea of Marfan is you do see that the legs would be longer in proportion to the torso. So it's not your whole body's bigger. Your limbs are bigger than they're supposed to be relative to your size. So I don't know. When I think of gigantism, I just think that you're just really big and everything is big. I don't know if that's what you're thinking about
Yeah.
when you think about gigantism. I don't know nothing about gigantism. I guess gigantism. Gigantism? Is that the opposite of dwarfism? Or is that being extra big or extra small? I don't know. Last example of variable gene expression is in this principle we call penetrance. Penetrance is basically the same thing as variable expressivity. It's just typically when we think about penetrance, we are usually talking about some type of disorder. And we think about how often a particular genotype results in a disease in a population. A lot of times penetrance is used with different types of cancers. So when they say you have a 60% chance of breast cancer, that's a penetrance calculation that was done previously, based off of a genotypic analysis. So we can look at how penetrant a genotype is based on how often it's associated with the disease. So that's one of the problems with breast cancer is you could have the breast cancer allele, but it doesn't mean 100% chance you get breast cancer in your lifetime. This means you have a likelihood that's greater or less than somebody else. Now there is a calculation that we are going to perform with penetrance, so it's a very basic calculation. It's going to look like it's complicated, but it's really not. And we're going to save it for Wednesday next week. And what are we going to do on Monday? Not coming to class.